The overall goal of the experiment demonstrated in this video is to assess whether the antimicrobial peptide catheliticin plays a role in the amelioration of dextrans sulfate-induced colitis in mice. This is achieved by injecting bone marrow from wild-type mice into irradiated cathelicidin knockout mice, and bone marrow from knockout mice into irradiated wild-type mice. The transplanted bone marrow will generate new blood cells in the recipient of donor genotype. The recipient mice are then exposed to dextrans sulfate to induce colitis. After 4 weeks, successful transplantation is validated by flow cytometry, and colonic tissue damage is assessed by histology. Results are obtained that show colitis is ameliorated when wild-type bone marrow is transplanted into knockout mice. Conversely, colitis is worsened when knockout bone marrow is transplanted into wild-type mice.
This method can help answer key questions in determining the gene expressions from bone marrow-derived cells or non-bone marrow-derived cells in inflammation or infection.
C57 Black 6 wild-type and cathelicidin knockout mice are recommended for this experiment. To make it possible to track the wild-type mice and knockout mice donor cells after bone marrow transplantation, use CD45.1 mice to represent wild-type mice and CD45.2 for the knockout mice. The chart shown here describes the experimental setup. 5 donor mice